Plotkin would develop a rubella vaccine to protect a new generation. His first order of business? To acquire a strain of the virus.
The virus was isolated from one of the fetuses that were aborted at the mother's request because she had acquired rubella. And indeed, when we obtained the fetus, it was clear that the virus had damaged the fetus.
Dr. Plotkin's lab alone received material from roughly 35 pregnancies terminated because of rubella infection. His RA273 strain of rubella virus came from the third organ, the lungs, of the 27th damaged fetus he had received during the 1964-65 outbreak. To make his vaccine, Plotkin would need cells in which to grow the virus. Cells are like tiny factories, making proteins and enzymes needed by the cell and by the body. A genetic code, or DNA, provides the blueprint, instructing the cell's components on what to do. Viruses also contain a genetic code, but they can't make their own proteins, so they need to hijack the protein-making machinery of a cell to reproduce. Once inside a cell, the virus releases its genetic code, which takes over the protein-making machinery. The cell follows these new instructions and becomes an assembly line, generating thousands and thousands more virus particles. These new virus particles are an exact copy of the original virus, and soon leave the cell so they can infect other cells. In the 1960s, researchers had typically used animal cells to grow viruses for vaccines. But the head of vaccine development at Merck, Maurice Hilleman, had recently discovered something frightening about animal cells while working to improve Jonas Salk's polio vaccine.
What Dr. Hilleman found was that the monkey kidney cells that were used both by Jonas Salk when he made his vaccine, or Albert Sabin when he made his vaccine, were contaminated with a monkey virus. It was the 40th known monkey virus of man. It was called SV40, and he found out soon enough that it also caused cancer in experimental animals.
This made people realize that cells from animals, as primary cells from animals, could be contaminated with a variety of so-called extraneous agents, that is, viruses that you don't want in a vaccine.
The challenge for Plotkin would be to find a type of cell that didn't carry the risk of contaminating viruses. It so happened that across the hall at the Wistar Institute, Dr. Leonard Hayflick had been experimenting to better understand how and why we age. He did it using human fetal cells.
Human diploid fibroblast cells are clean. That is to say, coming from fetal tissue, they have no agents that you don't want in your vaccine. So I decided that in order to avoid any problems of so-called extraneous agents, that I would use human cells for the attenuation.
Hayflick had received his fetal cells from Sven Gard at the Karolinska Institute in Sweden, the organization responsible for the Nobel Prize. Gard had obtained the fetal tissue from an elective abortion performed in the early 1960s, a fact that would later cause controversy. Because he was at the Wistar Institute at the time, Hayflick named his line of cells WI-38 cells.
A cell bank had been established at Wistar so that one didn't have to cultivate any new cells, but one could go back to the freezer, take out an ampule of the cells, grow them up, and then make a vaccine lot in those cells. I started to use my experience with polio vaccine, actually, to attenuate the virus, meaning, of course, to weaken it to the point where it would still immunize but would no longer cause disease.
Weakening viruses by attenuation is based on the principle of selection. Like with evolution, survival of the fittest is dictated by one's environment. Change the environment, say from the human body to human cells in a petri dish, and you suddenly change the whole game. Now, if you lower the temperature of the petri dish below that of the human body, all bets are off. The vast majority of virus particles will simply cease to reproduce, but a few will find they can adapt to this new environment. By passing these select virus particles over and over through the laboratory cultures, the virus population is soon made up of only those particles that can grow well in the petri dish, but can't grow well in the human body. Plotkin's temperature adaptation in fetal cells proved to be an efficient and effective means of attenuating his vaccine, and the work showed great promise, but not everyone thought so.
This was a new concept, and there was great difficulty in getting the regulatory authorities to accept the idea of using those cells to make vaccines. The attitude was, better the devil we know than the devil we don't know.
Plotkin couldn't have predicted just how much resistance his use of fetal cells would face, not only from the regulatory authorities, but also from his fellow scientists.
At the time, there was a dogma that human cells in particular could not be passed numerous times, or if they did, they would transform to cancerous type cells. But Hayflick and Moorhead showed that indeed the human cells would replicate, would multiply a limited number of times, and then they would literally senesce, that is, they would grow old, and the cultures would stop, would in effect die.
A meeting about rubella vaccines convened at the National Institutes of Health, and Plotkin was part of a minority advocating for the use of human fetal cells. Among those against the idea was oral polio vaccine developer Albert Sabin.
He got up towards the end of the meeting and made a statement about why diploid cells should not be used. But he had no evidence, and I remember very clearly saying to myself the biblical phrase, the Lord has delivered him into my hands.
Plotkin addressed the meeting, pointing out that Sabin's statements were not based in fact, and that all the evidence showed fetal cells were indeed safe.
the audience gave me an ovation. Now, that was not for me personally, but simply because scientists in general don't like ex cathedra statements, and it was clear that most of them had been convinced that the prejudice against diploid cells was not founded.
If his scientific peers were beginning to come around, those in the religious community were still grappling with the morality of using human fetal cells, even when developing a life-saving vaccine.
Indeed, some controversy did arise because the cells that were used to grow the virus were obtained from a normal fetus.
This fact raised questions among anti-abortion advocates and ultimately drew the attention of the Pontifical Academy for Life, then headed by Cardinal Joseph Ratzinger, who would later become Pope Benedict XVI. While the Academy weighed the heavy toll of using cells from an elective abortion, Ratzinger ultimately cleared parishioners to use vaccines developed with fetal cells to avoid the devastating dangers to human health, especially from rubella.
Their consensus view is that as the cells were not derived specifically to develop the vaccine, and inasmuch as one is not doing new abortions for every vaccine lot, one is going back to cells that were frozen in the early 1960s, so that no immoral act is taking place according to those theologians. Now, I'm sure there are other extreme points of view, and from my point of view, personal point of view, having seen the suffering of families with congenital rubella syndrome infants, I consider that what was done is 100% moral.
This study took place at the Whistler Institute, correct?
Yes.
You were at the Whistler Institute, correct?
Yes.
How many fetuses were used in the study described in this article?
Quite a few, but my answer to the previous question was, what did I use to make vaccines? And the answer was 2.
In your work related to vaccines, how many fetuses were involved in that work?
There were only 2 fetuses involved in making vaccines. When fetal strains, fibroblast strains, were first developed, I was involved in that work trying to characterize those cells, but they were not used to make vaccines.
Wasn't the purpose of this study to help develop a human cell line or to support the use of human cell lines in the creation of vaccines?
The idea was to study the cell strains from fetuses to determine whether or not they could be used to make vaccines.
So this was related to your work?
Well, yes, in a sense,
To
yes.
vaccines, correct?
Yes. It was preparatory.
So this study involved 74 fetuses, correct?
I don't remember exactly how many.
Turn to page 12 of the study.
Yeah, 76.
76. Mm-hmm. And these fetuses were all 3 months or older when aborted, correct?
Yes.
Okay. And these were all normally developed fetuses, correct?
Yes.
Okay. These included fetuses that were aborted for social and psychiatric reasons, correct?
Correct.
What organs did you harvest from these fetuses?
Well, I didn't personally harvest any, but a whole range of tissues were harvested by co-workers.
Okay. And these pieces were then cut up into little pieces, right?
Yes.
And they were cultured?
Yes.
Okay. Some of the pieces of the fetuses were pituitary gland that were chopped up into pieces too. Mm-hmm. Okay. Included the lung of the fetuses?
Yes.
Okay. Included skin?
Yes.
Kidney?
Yes.
Spleen?
Yes.
Heart?
Yes.
And tongue?
I don't recall, but yeah, probably yes.
So I just want to make sure I understand. In your entire career, and this was just one study, so I'm going to ask you again. In your entire career, how many fetuses have you worked with?
Well, I don't remember the exact number, but quite a few when we were studying them originally, before we decided to use them to make vaccines.
Researchers need a range of reference sequences that encompass the genomic differences in any ancestrally diverse population. Put together, they would showcase all human genomic diversity. Unfortunately, up till now, most data about genomic variants that may be involved in different diseases have been gathered from people of European ancestry. But that's just one ancestral group. This means we may be making some medical decisions for other ancestral groups based on incompatible data. But scientific efforts are underway to eliminate this historical bias. One such effort is the Human Genome Reference Program, funded by the National Human Genome Research Institute. Over the next couple of years, at least 350 high-quality, gapless human genome sequences will be generated from ancestrally diverse people. Remember those gaps in the genome? Another group of international scientists called the Telomere to Telomere Consortium are working to fill them in and generate totally complete sequences with improved technologies. Using these 350 complete genomes, the Human Genome Reference Program will then combine the data and create a unified genomic representation of the human species, calling it a pan-genome. Will this be enough to serve as a single reference for all human genomics research in the future? Perhaps not. But so long as these people are thoughtfully chosen, this effort will go a long way towards that goal. Slowly but surely, we are moving closer to one of the fundamental aims of the Human Genome Project. a complete and accurate representation of the genomic blueprints of the human species.